wrote down. What?
Oh, you can't find mine?
No, I can't find yours, but I don't want to mess up because I'm like, shaking people. So I don't want them to. That's it, right? Or are we missing that? I had one too, right?
Yeah, I need my strip and you need your plates. What happened? Your job?
No.
My brother.
Oh,
Like I was like prepared for that. But I wasn't mentally prepared for that. Was like negative something.
Not so bad.
I did very.
I can imagine. I'm alright. I had to email her that she had to quit. I was like, girl, I left all my blank, all my open responses. All my open responses. And then I guess it's a choice.
What'd you write? You wrote.
I'm sure you could feel my energy because I was pissed off.
You wrote Jenny on it. J-I.
Should be, or it's JP. JENNY.
And they're just like, it's not fair. You get Oreos. I will never
But
have
she
Oreos.
drops the lowest fifth grade.
Hi, ladies.
Mine went from a 98 to a 96. I got 35.
Where your stuff is, don't look, otherwise I'm just gonna lay it all out.
Hi.
You did your hair.
I cut it, yeah.
Cute.
I like Britain did your hair. I cut it right
I love
before,
it.
right before I left on Sunday.
That's why it's faded. I couldn't really see.
Oh. I love
You look beautiful today. Was that the hair for the vacation? That was the hair for the.
Oh, it wasn't vacation. I wish it was vacation.
That was for work.
For
It
the.
was work.
But that was the.
Yeah, I had to cut it. I needed to color it too because I have roots.
Looks pretty.
Thanks. I cut it myself, guys.
You cut it
And
yourself.
I color my own hair. Yes, I do it all myself.
You styled it and just.
That's why I don't do red anymore. I used to do red.
You went to school for that?
Yeah, I used
Where
to
you
do
just-
red all the time and I stopped.
What? That's so
over it.
cute.
That and blonde, this blonde that I got left in my hair, once it grows out,
Look at the
I'm
layers,
embracing the
though.
grays.
Look at the layers.
Mm-hmm. Yeah, I'm embracing the grays. I'm done dyeing my hair. My hair's already
Well,
falling
whenever
out.
you want to, I'll take your appointment book.
The
You
girls
do
at work
hair?
have me locked.
She just said she
You-
does
Oh, you do hair. Okay.
I don't do it
as
I
a
used
job,
To,
but she.
no, I used
But
to
she
before
can't.
COVID. I did. I used to do makeup for weddings and.
There you go.
You went to
hair
school for it?
weddings.
No, she
No?
said
Okay.
she just has a hand for it. She said.
But then COVID happened and it ruined it. You know?
I did nails like a mofo when COVID hit.
Did you?
I made mad money.
Yeah, I'm sure, because the salon.
We're all closed.
Okay, so we have a few things to do today, so I want to get going. If you don't have your material yet, I'm just gonna shout out initials. This is yours. DTP. AW. AM. No?
AM is me.
You can't even see the dark ones to take pictures.
Dude, is that
AM. This is another AM. This is yours.
I would have rather been doing it. Trust me.
That's what's all mixed up.
Those,
What's your blood?
Which one's that?
Right.
Ooh.
I think that's our combined.
Yeah.
Wait. You got a blue one?
AD.
Hers
then
turned pink.
also-
That turned pink.
Which one is that one?
This is HEA.
HEA.
MCO.
Oh, mine's orange.
This one turned pink too.
Is that HEA?
This is.
Or is the pink one HEA?
This one is
Oh, this side.
MA.
That was HEA.
This one's MA.
For all of them.
Is that HEA? Yeah, Mimi's turned orange too.
Oh,
Mariana.
I just
Hers,
realized
hers turned orange too.
if there were new ones.
Yeah,
Which one's that?
yeah.
E,
MN and YC.
EMB?
MN.
What do I see?
You were labeled without you knowing it. Okay.
You said EMB?
Oh, and then Dan, your strip is over
What
here.
is it? I don't know, which one did you just
And then
show
there's
me?
a strip here.
Oh, mine's
Someone
green.
see your strip? I can't wait to
That's this one. Oh, hers turned.
This goes with this.
You guys, your guys
And
turned
then
the
when it
same.
gets
Hers
time
turned
to
lime
do
green.
this,
Like
That's
chrome.
cool.
58.
Okay, thank you.
That's like a chrome color.
Yeah, they're pretty.
If so, something is wrong. Is something wrong?
What did your SSA?
SSA?
Okay.
Yeah, it looks like it could be AM. Okay, so before we get started, just a reminder, your report on your identification of unknown using the biochemical test or
MAG.
extracellular enzymes was due today. Hopefully everyone submitted
MA.
that to Canvas. You have 3 things due tonight. Chapter 10.1/11 study guide, concept check, and homework are all due at the end of the day. You have a virtual lab due on Tuesday, effectiveness of antibiotics, also known as the Kirby-Bauer
Are they
method.
supposed to
That's
be
due
long?
Tuesday by
Long?
2.
Or
Next week, just a heads up.
No, how did you
there's
draft
3. Okay, this is on chapters 8 and 20.5, and then 10.1 and 11. And then finally, the handout
MAG.
with the results from today's lab. Your identification of unknowns for enterics is due next Thursday, a week from now. Okay? We'll go over that paper, so there were some things here in the middle bench. Everyone should have collected this, just so that you know what is due in 1 week. There's all your results for today's enteric lab. Then these 3 things are for the enteric identification. You can pull one of these papers off, and then these 2 laminated sheets. Okay, so everyone should have
What is
that.
your MAG? MAG.
We are going to start with the conjugation.
No, MA. Yeah.
So put your
Hers is pink too.
colorful
Your guys is like
media
similar.
aside. And we're just going to find those plates that were LB amp, LB strep, and then LB amp and strep.
Yeah. Just look at it one.
Okay. So who can tell me very quickly, very briefly, what conjugation is?
The horizontal transfer of NA from one factor to
Yep, and if you were to differentiate this method of horizontal gene transfer from transformation and transduction, what does this one require that the others don't? Does anyone remember? I have terrible hearing, so you just gotta keep it. Huh? One of them has to have that plasmid, but also what? What else?
Like the 2?
Yeah, so that conjugation to direct contact. Remember, the cells had to come in direct contact with each other. So the F positive cell that had the F plasmid has to make contact with the F minus cell, the cell that lacks the F plasmid. And then through that conjugation tube is where it transfers one single-stranded copy of the F plasmid, and then once it gets there, a second strand is made, and then the second strand of the original is repaired. So in the end, we have 2 F positive cells. Okay, that's the idea. So if you remember, we started by inoculating plain LB media with strain 1 and strain 2. All we knew was that one of these strains has an ampicillin resistance antibiotic, the other one has a streptomycin resistance antibiotic. Streptomycin resistance gene, sorry. Okay, so we started by just inoculating plain LB media. LB media should support any kind of bacterial growth that can grow in the laboratory. Okay? So we did a single line on each side. Do you have growth?
Yeah.
Do you have growth of both?
Yes.
Okay.
One of them's missing.
So we're expecting growth of both.
LB?
So
Oh,
why do
just
we even
LB?
bother doing this?
Oh yeah, just
Yeah, I'm
To
confused.
make sure that we have a control. Like,
Yeah.
This is where they grow just without anything going on.
Yeah, to show us that we have viable cells to start.
Oh.
This comes in really handy to troubleshoot if you don't get growth at all. Let's say all 4 of your plates you have no growth, okay? And you look back at this one and you say,
Means
okay,
it
well, if we
required
didn't
something.
even get growth on LB, then something's wrong with our cell line, okay? But if you got growth on your LB and no other growth, then you can start kind of figuring out, okay, well, our cells were alive to start at least, and then something else happened.
Yeah, that's
Okay, so this is our positive control to show us that we have viable cells. How about for the LB that has ampicillin in the agar? What did you guys get?
Is that the A?
LBA.
A.
Growth of only strain 2. Is that what everyone got?
Yeah.
I got growth on both.
Got a little growth. on
Got
one.
growth on both.
Okay, I'll come and look at it. Go ahead. Everyone else just get growth on 2?
I see what you got.
Anyone?
Is this the one we're looking at, right?
Oh yeah, you
Is
did.
it just something
That's
small?
No.
This
one
one
we
is the
didn't
only
get.
one we didn't.
It's also here. On the
Oh.
I have no idea what.
Okay.
That is.
But this to me
here
looks
too,
like it's
look.
not E. coli.
It's not E.
right?
coli,
It's
but
some
it's.
kind of
You got a little bit of growth.
Unless it
Yeah,
was
yeah.
labeled.
I just find it
Because
interesting
we
that
got
it's
these
into-
labeled.
It's
Yeah,
odd
they
when you
should
can
be
say
labeled.
it's
Yeah.
not strain 1.
Oh,
It's not
okay.
strain, that's
Yeah.
interesting.
Okay, so if you see growth
It's not
of
supposed to?
strain 2 on this plate, what can you conclude? What about strain 2? So
So do we
strain
ignore
2
this and just?
Are we supposed to?
has
We're not supposed to be writing anything.
an ampicillin resistant gene. The only way it can grow in the presence of ampicillin is if it has an ampicillin resistance gene.
I'll take a picture of it.
How about for the LB plate with streptomycin mixed in?
Ours didn't.
AS. She said the AS.
Just growth of strain 1.
We only got growth a little bit of strength too.
And so what can we conclude here?
Not
I can't see. We got growth in both. I'm so.
Are you confused on like how we're getting?
So,
On both.
if you have growth of just strain 1 on the LB with streptomycin, then what can we conclude?
That has resistance to streptomycin.
Which one? has
Strain
this?
1.
Strain 1 has a streptomycin resistance gene.
So, you see how BA, she
Yeah.
she
And
put
then-
next to it. Ours, I just wrote it down. It was the opposite.
We
'cause
had
got growth.
the
We're supposed
LB,
to have growth. So I'm just,
ampicillin,
I'm gonna take a
and
picture of that. But just know
streptomycin.
LB1. We were the opposite of that.
And did you get growth here?
For both, because the other one we weren't supposed to have
You
growth on
did?
both either,
You
she
guys?
said.
Very slightly.
Okay.
Like a dot.
Okay, So what do you think that we were hoping to see? What should we have seen? If everything was prepared properly, everyone, all the sections before you, including you, used proper aseptic technique, what should we have seen here?
No growth.
No growth. This was our negative control. That should confirm with us that conjugation has not occurred. That we simply have these two individual strains, each with just one antibiotic resistance gene. If you have growth, what that tells you is that either you guys or previous sections didn't follow good aseptic technique and somehow mixed the two strains. So imagine that when you went to get strain 1 and you inoculated it, maybe you or someone else didn't fully sterilize the tool, mixed it into strain 2, introducing 1 into 2. And now you're allowing conjugation to occur in that tube. Okay? Which would explain if you got growth in both before. Okay? There was probably some contamination of that stock culture. And again, remember before us, there's one other section, no, two other sections that use those same stock cultures. So we're relying not only on our own aseptic technique, but on theirs as well. Okay? So you should be able to troubleshoot your own, to say what can explain this hypothetically, but also know what to expect in an ideal situation. Okay, so then we had our mating plate. And if you remember, we took a loop full of strain 1.
Did you line them up
Put
like
it in the
that?
middle, then we sterilized our loop, hopefully. Grabbed a loop full of strain 2, and just mixed it throughout. And so we just might just have this blob in the middle. Do we have growth there?
Yes.
Does that tell us if conjugation occurs?
Not inherently.
No, it doesn't tell us anything, because think about it, even if we just took one strain and did this, we would still see growth. Right? This doesn't confirm anything. So this is where part 3 of conjugation comes in.
Not yet.
Now, the true test of whether conjugation occurred is we now have to take some of that growth from the mating plate and we inoculate LB amp, LB strep, and LB amp and strep. You do not have to separate it in the middle anymore because you're grabbing from this mating plate. Because remember, we are hoping that through conjugation we created essentially strain 3 that has both ampicillin and streptomycin resistant genes. Okay? So now the plates, we have so much material, it's in the back.
Yeah.
In the back, each group is going to pick 3 plates, 1 LB amp, 1 LB strep, and 1 LB amp strep, and simply inoculate each with just a line, it's fine, taking from the mating plate. On Tuesday, if we see growth in all 3, we will know that conjugation occurred. These are pretty much controls. This is the one that we're definitely looking for. If conjugation didn't occur, we wouldn't see growth in this one, right? That's what we're looking for, a cell line that has growth. Okay, so go ahead.
Oh.
There's no way we should do it, so you're just gonna have to walk through the rest of the steps.
So do you get what she said about like the accidental conjugation happening because of contamination?
Yeah.
Okay, cool. So you're still
So
gonna,
probably one of us, like, we
you're
probably
still
did it
gonna get almost the same exact results on this one because your bacteria already had sex.
crossed, yeah.
Yeah.
And it's both because this one's greasy.
Yep.
Okay.
Yeah. What this tells me though is that your strain 1 got this strep, resistance, but not. But not the ampicillin resistance, because otherwise you have growth here. So. Yeah, which is accurate. So it looks like your strep didn't transform, but your straight tubes did transform. So it was probably your stock culture that was contaminated, because otherwise you would have consistent growth across all of them. If it happened on your plate, it was likely the stock, which means it wasn't even your fault.
So whatever we were pulling from.
Yeah. Yeah, the contaminated stock, because otherwise you would have, these guys would match.
Right.
Does that make sense?
It would be growth on
Yeah. That means it didn't happen on your plate or on your loop.
Are we gonna inoculate with a needle? Or
So that's what I was gonna ask. Do we inoculate with the loop and Anita? We're just scraping and then putting on one streak line on that.
One
correct?
streak,
Is what I-
one streak from that plate on each.
With your
Okay.
loop.
with a
Yeah.
loop?
With the loop.
Yeah.
For the sterilizing.
You only use the needle
For
for
stabbing.
slants. Yeah.
Yeah, stabbing or slants. Or stabbing. I forget what it's called.
Yep.
Deep inoculation. I've only done it deep inoculation once, so it's deep inoculation.
There's two.
So it must have mixed streams. So that's why we're gonna put like. Are we gonna be breaking this?
It would be probably good to write your growth note
Yeah,
down.
just
growth.
missing or broken?
Or if you're okay with your pictures.
Yeah, so
if
it
you think
should
you
be
need to
more in
organize.
that glass cabinet in the corner,
There's also,
under
you
the
could
first
create
aid
like a chart
kit.
like this if you wanted,
Yeah.
but obviously
Oh,
with different results.
Did you want me to do this?
I forgot to do this.
It's okay. Then you can take, it'd probably be good just to take your photos with you or something. Yeah, if you don't have the paper.
Okay.
I took the last loop that wasn't there.
Thanks, Glenn.
I don't have a loop either, but we can just do it.
Okay. I don't mind lighting it and watching.
I don't want that back.
It's burn. Yes. She said, "Let me burn it."
That was such a big laugh, yes. What are you doing here? So I can do it?
I like to watch and look at it.
I'm doing it hot because I feel like there's like a little chunk on there. There is. Mine has a chunk. Well, this one's like new.
That one's brand new.
Like in the,
Loop is not even.
right, yeah.
There you go.
Not the steroid shots. Or you can get Botox or steroids.
As you go.
We have to sterilize in between the months.
No, Botox is
Yeah, I'm assuming because we gotta pick
depends
up.
on the
for each.
severity of their migraines and how often they are.
You see?
We
Not
sterilize every single time we inoculate, correct? Okay.
My
Can I use your crayon? I stole everything of yours last class
Here.
too. Oh, she gave me hers. Like my drawer was.
So you can have this one, cause there's two in here.
Okay. Mine was like empty, so I was like, "You didn't see us?" I said, "This is no." I think I deleted the first time because I ended it, but I said, "This is gonna be yes today." I said, "Everyone say hi to yes."
It's been bad with the storm just sitting.
Today's the 29th.
Today's the
If you guys
30th.
are done on that handout, you can start recording your cultural characteristics. At the top here. This one? Yeah, this one. And this was from Tuesday.
Thank you.
You need help?
Yes.
You just look a little lost.
So once you're done with your 3 plates, put them in the incubator, please. Conjugate.
Talking shit.
No, I'm laughing
Once
at
you have
Mariana
your
because she's moving her chair. It's like,
Plates, like either photos or whatever results you want written down from your other plates, you can throw them out. Not the colorful ones, plain ones.
These?
Yeah,
Okay.
You did that one though?
not the ones you did
have your
today, no,
colors?
either.
You have the pictures of these already?
did I take pictures
Yeah, you took pictures, and this is, you took pictures of this too?
Yeah, let me just double check this.
Okay.
So we just need pictures of them and that's it?
Yep. So that way you can write growth, no growth in your experiment,
But because
in your
this
lab
is
notebook.
mixed together,
Yeah.
it
So we don't have to explain it or why we didn't have to?
Yes, you kind of do. You should, but like you have notes on it, like the
Sort of.
improper sterilization.
I kind of need help so I can get them better.
Okay.
Because I just want to make sure. What happened? Oh, snap.
Center chair
I just want to
today.
make sure that so. LDA, so I would put a stream
so I
to
can
have
take
an
pictures of
AMR
this.
Oh, your
gene.
negative control show good?
She did it,
Yeah.
Yeah, that was nice.
Uh-huh.
So once you're done with your 3
Oh,
plates,
yes, that makes
put them
sense.
in the incubator.
So LBA is the opposite of what she wrote up there. So it doesn't have an
You can start
AMPR
with the
gene.
intergenomes by
So
recording the
LDA
cultural characteristics.
Is going to be
As soon
your
as everyone's done with
ampicillin
the
plate.
conjugation, I'll go over the
So,
selection. I
strain
think this is
2
overflow, so.
was supposed to be ampicillin resistant, but that was not true for you. So your positive control was actually the LB plate, and that's normal. That just shows you you had
Because I'm like,
viruses.
I don't want to get home.
Yeah, you can totally steal the chart. And go
We
for it.
forgot to bring it.
It's. I honestly don't know if you guys got this.
We did.
Or, oh, okay.
You want the one on the bottom of
Close
it?
it off.
Yeah.
Hmm.
Yeah, there's no.
Or yeah. You have a daughter?
That's my son.
That's your son? Oh,
He just has
I
really
like those
long
little
hair.
braids.
Yeah.
That's so cute.
Everybody thinks he's a girl.
Honestly, children all look genderless. It's just all in the hair. They're just round little things. Yes, with your observations. And if you need to
Yeah.
compare, there's also the blank ones from last
Paper
time
thing
over
with
there.
the form.
Mm-hmm.
Give me
So-
1 second.
But she's also got
One.
slides.
No,
So for my,
thank
for my LB, it was growth.
Yes,
That's
on both of them.
good.
Growth, expected.
Okay, so I think everyone's done, right?
Mm-hmm.
So,
I can come over here.
you are going to record all of your results from your plates on
Should have been
this
no
form
growth
here,
and straight.
okay?
So
We are going to start by going over
Growth on
the
both.
4 selective and differential measures. Whenever you have time, make sure you record the cultural characteristics from your
Yeah.
TSA. So, first up, EMB auger. EMB auger. I have background on the handout I gave you. That EMB auger is selective for
No, I do my picture.
gram-negative bacteria. So, the first thing you would write here is there growth? Yes or no? If, then it says, what can you conclude from the presence or absence
one?
of growth? If EMB is selective for gram-negative bacteria, and you see growth, do you then conclude it's gram-negative or gram-positive?
I mean EMI,
Gram-negative.
EMB.
Okay, so that's what I'm looking for in this column.
So,
See growth, and your conclusion is that it's gram-negative.
Mm-hmm. And it's clear.
You were then going to describe any color changes for the colonies and then the media. And note that this information, what I'm looking for in that last column
No.
there, is this information will tell you about whether or not you can ferment lactose.
The presence or absence.
So,
The
if
pink
you,
line on the-
if your microbe is
pink
not a
line.
lactose fermenter, then your colonies will more or less be clear. If it's a moderate lactose fermenter, then you typically typically see either pink or dark purple colonies. If it's a very strong lactose fermenter, then you see this beautiful metallic green sheen.
Which means that it's not
Yeah, so if you don't have one at your table, you should let me know, because
So it's not
it's
lactose.
worthwhile looking at, but also just to see the variety of results. So it can be so.
You said it's not lactose.
fermented.
So where does she want us to write that in?
On this paper.
You don't put like this, the second box?
You have a shiny green, don't you?
I think so, yes.
You see the shiny?
Yeah, it's like chrome.
The fact that this
Describe any color changes from the other. Yep.
That one.
How are
doesn't
you
change
guys?
color.
Mine's
Actually,
green,
at all.
so I was gonna take it.
Where's your screw?
So you
Oh,
want
it's in
me,
your plate.
so you want me to write it here?
The fact that there's growth
Yes.
tells you it's gram-negative,
Oh, okay, okay.
okay?
Um, since this one isn't a differential for the color of the agar. Sorry, I don't think she realized this one.
And then describe color changes is when we put the
Like-
green or red or.
Like the
Yep.
color
So
of
it's
the
a color
colony.
change of the colonies, and if there's color changes of the media.
Okay.
For this agar, the media doesn't
You
change
would
so
just say
much. But with the
No
other
lactose.
stuff,
fermentation. Yeah, it would be the same because
just to show you,
you have no change at all. Just weird.
just to show that's the strong lactose.
So no lactose fermenting for this. It doesn't change color, same thing, no lactose.
Yeah,
What can you exclude from any color change?
Eosin,
that's what I'm saying. It's the no lactose fermenting. So just extend your arrow.
So
I
is
guess.
everyone clear on how to fill that out for that first line? So if you have growth, do you have growth? Yes. So you're putting yes.
No lactose.
That tells you it's gram-negative, because it's selective for gram-negative.
So
This media is also
it's gram
differential.
negative.
So
You can put that note here
And for describe color change, I put
in the
green sheen,
But I'm sure where.
Right here.
So for this one, it's just.
and then
Because it didn't color change.
No, because it grew.
Yeah, this one.
Yeah, these are all selected for gram negative bacteria. Yeah, no, it's okay.
Describe any color changes for the colonies. So I don't know what they look like.
So your McConkey
So
is gonna be one of these two.
how do I know?
That one, yeah.
So before it was just.
Yep.
It should have been an
It should have been MAC, but that's okay. You know what it would.
like a little what?
Process of elimination. Alrighty. Great. So
Yes.
it's gram negative selector again, so that means gram
Okay,
negative.
next one,
Mhm.
So can grow.
McConkey agar. This one is also selective for gram-negative bacteria. So if you see growth, then that middle column there, what can you see from the present job is the growth? If you see growth, it's gram-negative again. Remember, you're using the same species always, so hopefully if one is gram-negative, they're all showing up.
You have some black precipitate. Yeah, put the lid back on.
You are now
And look at it
describing the
again.
color
Oh, wait, no,
changes
we have no color
of the colonies and the
Yes,
media.
Yeah. So no.
You can see here
Yeah,
that
no
the
lactose.
media is turning this bright pink on the left, as is the colonies.
Yes.
If it doesn't ferment lactose, you can see that it's more or less colorless. Sometimes it's that yellow,
Oh wait, sorry. You
sometimes
should put that here,
it's
and this
just
one
these
should say.
clear colonies. But what we were looking for as a positive result is this like pretty pink.
Because it's saying like what color was it?
Sometimes it's just the colonies, sometimes it's both.
It's okay.
Because like my column's pink, but my media is more like yellow.
But you're
You have the form from,
describing
Tuesday.
So this one
can't ferment lactose, which is great because then that means you're getting consistent results. You're McConkey? Oh wait, oh no, it's not.
Okay,
You're working.
so the fact that your, just pink colonies, that means that it's like a moderate lactose fermenter. If it was the very strong lactose fermenter, that's when the colonies are pink and the media is that right.
So the media did change though.
I ended up with two somehow, so.
So originally the media was red.
Yes.
So the media did change. chocolate yellow.
Yeah, it changed, but and you
And
can
then you
record that.
basically have all those
But
the
it's
results,
not the pink that we're looking
except
for.
for these two.
Yes. Okay.
So if you want,
So just to
you can
repeat,
get the
from
same
a
picture.
copy, if only your colonies are a slight pink, or sometimes just a little bit is pink, but the media is either yellow or
Okay,
very
that's the
muted
the
pink,
chip cake soy.
then that's
I think
moderate
that's
lactose fermentation.
Just, oh, that's your,
If the thing is
That's your positive
bright
control.
pink, like in the upper left on the left-hand side, then that is strong lactose fermentation.
so,
And if you see
That's
no
great.
pink at all, then that's no lactose.
And that's what you want us to put at the end on what?
Right.
At the end, the last one? Okay.
yeah, and she'll, no, sorry. No, because it grew, it's gram negative. Because they're all selected
Mimi still
for it. The change actually tells you that it fermented the product
Any questions?
and produced
I get the pink.
something.
All right, H and E
If
agar.
it's all the way,
The original
So then
agar
it's-
was this
if
green.
it's all the way pink, that means
If the,
strong.
if the microbe does not ferment
Lactose
lactose,
fermentation.
then the
I
colonies
have to put
are
fermentation.
clear.
Strong lactose.
If we see these yellow, pink, or orange colonies, then that just tells us that they're a lactose fermented. We can't see moderate versus strong, just lactose fermented. But H&E and SS can tell you 2 bits of information on the differential part. Whether or not they ferment lactose and whether or not they produce H2S.
So it's
So yours does not
So,
ferment lactose.
if you
It does
see
produce H2O.
that yellow, pink, or orange colonies, that tells you they ferment lactose. If you also see any black in the center of the colonies, that tells you they produce H2S as well.
was a color change.
Okay, so for
Yeah.
H&E and SS, that last column should have 2 bits of information. Whether or not they ferment lactose,
And then no
whether or
change.
not they produce H2S.
And then produces H2S, non-lactose fermented. No,
I mean,
H2S, yeah. You're good. And I think that's exactly the same case for
you said it's
yes. And it does not. Again, it says.
And
no
then the
lactose
last
fermented.
thing, the Salmonella, Shigella, Otter, SS, Otter. This one again is selective for gram negative. All 4 of them were selective
This
for
one,
gram
because
negative.
it turned black again.
So write down whether or not you see growth and your conclusions from that.
program.
Because it would turn right
If your microbe does not ferment lactose, then your colonies would be colorless. If you have pinkish red colonies in the media, then that means they ferment lactose. And then if you also see that black coloration, that means they produce
And no
H2S.
pen, so no change.
Produces H2S.
Any discrepancies or questions?
I have some colonies that are brown as well. I'm not sure if it's contamination or not. It's the same thing.
So it's black colonies, it produces H2S. And that's what this is.
So I would, this, I was gonna say that's orange, like HG.
No,
That's
yeah.
good. So basically, it doesn't
I
ferment
don't know if I can make
lactose.
a conclusion on that.
They're good.
You can
it's
be
black?
close, definitely, yeah.
I'll just note it down.
No, I
I didn't
thought
know if that was like
If
some
it's,
other
if it
like
has black colonies,
outlier.
it produces H2S.
Right,
Which means it lacks,
No, and it's
which
important
means
to
that there's
know,
no
I'm gonna talk a
lactose.
bit about like,
Oh,
looking at
okay.
any discrepancies, you had no H2S here, right?
Yes.
If you had yes H2S,
Then
and this
that's
one
what
didn't
this
turn black,
one
I would say like, but it had brown, so maybe
No,
that was.
there was no color change. There's no color change for. Oh, which one
Okay,
is
is
that?
everyone else set
I'm
on
not
that
even,
part?
I'm going like through it. Oh,
Okay.
okay.
So-
Cause for the,
You should have this
SSA.
whole table filled out. This is what's due in 1 week. The number one, were there any
What's the
discrepancies
background color
in
you
your
would say?
results? And
What
so,
color would you say the
look at
background
above.
is?
If one of your conclusions is that it's gram positive and another one is gram negative, what's the same species? That's discrepancy. If one of your results is that your
Metallic
microbe
green.
is a lactose fermenter and the other one is that it's not a lactose
So what
fermenter,
would you put the
that's
background color
discrepancy.
as?
Same with H2S. So they should agree. Okay, the other thing, again, just like with the extracellular enzyme or biochemical test, whatever your microbe ends up being identified as, I want you to just tell me a little bit about it. All right, so there's one more thing,
a TSA please?
bulky thing that we gotta do. And that's to finish with that API test strip. So if you have all the information from your plates, you can kind of push them aside. And we're going to continue on with our test strip.
That's the paper she had of this
You are
green one. I'm
now
just like
at
confused.
step 2 here, and we need to collect the following reagents. And these are the back,
I guess
back
I'm not
table.
understanding what I would still put the
You need to
background.
collect TDA. We're going to add 1 drop to the TDA cupule. You need to collect VPA and VPB. Remember, those are those little glass vials. And then you need to collect James or Kovacs reagent.
You said
Okay, so-
or?
Huh?
You said both or?
All, those
It's
are
all the
4
same
things.
thing.
TDA, VPA, VPB, and James, or I don't know, how is it labeled?
It's James.
James reagent.
Okay.
Okay, those 4 things, you can share them with your group. We're going to be adding 1 drop of each to the couple. Okay, so go ahead and collect those in the back bench.
So this would be dark
Moderate.
purple.
If it
So
was large, there
dark
might be less of
purple
a
colonies.
line.
Okay. They're strong fermenters. They're lactose fermenters.
For the media, we would put dark, because I got, mine is green, but like, for the background, if we're supposed to put background, I don't know.
Oh yeah, yours is green.
Yes, you're
Well,
correct.
what would I put for, what are you gonna put for background, because yours is like the same color as mine.
If the media started like that, there's no changes.
I don't know how it's done.
Look at the original one. What is she? So right here, look. It's asking you to describe any color changes for the colonies, right? So you have green. So change. Right.
put green.
So you put green and
Metallic.
any describe any color changes of the media.
There wasn't any color.
There
change.
wasn't any color change.
So that would mean that you have a
Yeah.
strong lactose fermenter.
Because the color changed for the color. But dark purple is also a fermenter.
Mm-hmm. Yeah, it's just a weaker fermenter.
That's strong because it's not. Damn, I need your book. I shouldn't.
Okay, so once you're at those
Margin means
reagents,
the
you're going to add 1 drop of
remember.
TDA to the
Form
cupule marked TDA.
resin.
So take the cover off, and then add that drop.
Yeah. Margin.
And then once you do that, you'll find the
I know she's
VP
right here with
cupule, and add 1 drop of VPA, 1 drop of VPB.
I would say
Remember, you may
irregular.
have to crack those open
Irregular for the form.
by bending them in half.
Yeah.
Irregular.
Yeah. The regular is a circular. Somewhere sorry.
And then for the
Low base.
just
1 drop
everything
of
else,
TDA
right?
to
Just kind of
VPA
figure out the
cupule,
margins.
1 drop of VPA to VP, and then 1 drop of VPB to
What's up? Which places? Sorry.
The MAC.
Oh, the McConkey?
Yeah.
Okay.
And I thought that there was no color change.
Mm-hmm. Yeah, I think that's the color change is kind of just what you're seeing is in the colonies. I don't think there's a change in color in
Okay.
the background
So that
because if there
means-
was, it'd be turning more yellow.
So it'll be like, would that be does not continue?
It does.
It does, okay. So if there's color change, it does not produce, does
Yeah.
not ferment.
So the McConkey, the agar will tell you if it's acidic. That's all that means. But yours did not change color, so it doesn't have acidic.
Okay, I know what thank you. I was confused on like so if there's color change that does not include
And then if you have that done, you're gonna add 1 drop of James reagent to the IND cup.
Yeah, so this does indicate that because you have pink colonies. So that does, I would say here, color change, pink colonies, because you can see that they're a richer color
I can.
in the
Thank you.
around. And that would indicate that it is a lactose fermenter, which is consistent with the other results, so that's good.
Okay, so
So you're going to add your reagents to these 3 guys here. You have your TDA, your IND, and your VP, which is the VPA and VP. And then I'll leave it.
little? Yeah, they're like
Honestly, your notes are good. I would worry about that like after.
No, not put it. I don't put the numbers on.
It's good to have pictures, but not required. I'm just saying that this has to take like a certain amount of time to take your pictures after you do that. Has to sit for 10
Oh,
minutes.
really?
Then if you're done with that, you can bring your TSA plate over and do a oxidase test.
I'm about to say fuck this shit.
Oh, and return the reagents, please. You're done. You don't mind waiting until people have bringing it back.
Oh, here you go. Here's some reagents for you.
Thank you so much. Thank you.
And just like, might be good to take a pause and do this because
We need
it does
these
need to
here.
sit in there for 10 minutes.
Oh, okay.
Yeah.
And you're
The
putting a
VPA.
drop of each and
Yeah, so that, do you have your
are we
protocol?
putting them in all of them?
Oh, that's your quiz, my bad. I can't read that far. I'm
Me
dyslexic
either.
and have bad eyes.
Which
So the TDA.
I'm like worried.
it's
You
TDA,
gotta go get.
the VP
Are
gets
you
both
gonna?
of
Yeah, we have
them.
those.
The James reagent goes in the
And for
IND.
TDA, IND, and VP, you're putting.
You're putting the VP in the VP, the IND gets the James reagent, and the TDA gets the TDA, which is the fluoride.
So I should only have 3. Why do I have 4?
You should have 4. Yeah. So the VP gets A and
drop?
part A and part B.
So TDA.
Yeah, TDA gets TDA.
1 drop.
Yep.
1 drop. 1 drop. Yeah.
Do you want to drop?
So we don't need a picture for this, right?
No. And also, it's gonna take a minute to react, and it's best to just have a picture of your end results afterwards.
TDA. Okay. And the James goes in the
Remember with the
IND.
oxidase
Yeah,
reagent, you're looking
James
for an
and IND.
immediate color change.
Don't take this.
Just the 1 drop.
The IND.
Yeah.
I know this is a bit of a bottleneck, and I'll go over this again in a bit, but you're gonna start by comparing your test strip to this top and
Do
bottom.
VPA.
Top is
1 drop.
positive,
And VPT.
bottom is
And it
negative.
should already be equal.
If it matches a positive
So 1 drop
result,
in each.
you're going to put a
And
positive sign.
1
above
drop
the
of each
sign.
in the VP.
Yeah.
Oh, sorry, I
It's
thought
a-
you
if it
pointed
matches
to
a
each.
No, no.
negative, you're gonna put a
No.
negative sign there. Oxidase test result goes over here. You'll see one that says OX with dotted lines. Positive or
And they wanna
negative.
come out.
Yeah, and I'm just gonna give yours a little tilt because I noticed it might go all the way. There you go. That's how you can make
That's
sure
for
it's a little
VP.
mixed. And if your drops kind of sit towards the top, you can do a little tilt. Yeah, that's Mari's got it. Cool.
That bubble there.
Both of them in VP? Yep, 1 little drop in each. Did yours goes down when you tilted it? Not really. No, right? I'm not going down nowhere.
My IND turn right.
Yeah,
Cute.
And then
But any change.
it looks like it has.
Okay.
Any change.
Like it looks like clouds in the VP for mine.
I was gonna say, like,
Maybe it's your phone.
am I supposed to mark out, like, negative?
Any change.
Alright, so form was entire, I think.
And if your result.
I was gonna say, I think I ended up with an air So
Yeah.
it's like, it's changed, but there's also February.
Yeah, mine is like that too. Oh, okay.
Yeah,
Mine,
that
yeah.
makes it kind of hard to result.
What did you say, 10 minutes? Yeah, it's gonna take a bit to.
Yeah. Slightly more orange. Yeah. I would say any change at all is a
And
positive.
then we need to do
So
the
it doesn't have to
oxidase
match
test
that
on the, where are we putting these results on? Here, right?
There is for like your mind.
Oh, there's
But
an
for the
air
whole-
bubble found its way in there, but
Usually
it
they actually
has
run this
changed.
through like,
It is a pink issue, so it's pretty clear that like if it had.
Yeah, and it's just like.
I knew that I ended up with a very tiny air
So
bubble.
just a
in
plus
there,
or
but I
negative.
Okay, so then for example, you are not yellow.
It's automatic.
Okay.
If it's not yellow on the left side, it's not blue on the right. Just breaks it further.
It's good to know. 'Cause I was gonna say, one of mine, my
Glutamine is turned brownish olive instead of a yellow.
Yeah.
That's also not.
So if there was no change in the
So-
media for EMB, there's no lactose ferments.
If there's no change,
Yeah, in EMB, because I have no change.
Yep.
And so there's no lactose ferments.
yeah, no lactose fermentation, no lactose fermentation
I would say that's close.
across the board.
Oh, that's really
And just remember.
She had a, oh, she did have a change.
Yeah, this one was orange, so it changed, which means
Oh, oh wait, so she
Something
had
like
the,
that
oh, I
with
thought you were
the
asking me about
metallic
yours. Okay, so she did have the metallic green.
Yeah.
So that does mean it does.
Does.
Lactose fermentation, just erase the no, I guess. And then,
orange is change.
Do you know what
yeah,
to do?
orange is changed
Lactose
Okay.
ferments.
So you're gonna use this.
to compare. Yeah, because it looks like your VP already reacted, so you're good to go.
And so,
Mm.
this is yours?
Yep.
Okay, you start with your ONPG
And then pink
and try to
is
match it to either one of these.
lactose
Yeah.
fermentation.
This
if-
looks closer to here.
If
Right,
there's
is that
any
kinda
change,
yellow?
I've been telling them positive.
These are all positive results. So now you put a positive in that little bubble.
Mhm.
Plus, yeah.
Wait, if it grew, it's gram negative.
So
If it grew, it's gram negative. She's got that
Oh, she
noted,
got
she's
it.
good.
Okay.
Yeah, you're all good.
Okay.
All right, I did
Then the
all
next
of
one,
these.
you match to either one
And I
of these.
think-
And you,
People's
Okay.
VPs are changing a
If-
little faster than last
if
list, so I
your
think you should be
results
good.
don't quite
start
match
marking.
Yeah,
either
like
one of
this
these.
one.
What is it?
This one looks like it has less than this.
Oh,
Or
the
that's
amount,
okay?
that's okay, yeah. But if it's, I
So
can't-
if it's any of these, it's positive.
Yep,
If
this
it's
is
any of
negative.
these, it's negative. So
If-
in here, we're putting positive or negative.
Correct.
So before you walk away, this pink one would be up here.
Yep, and you can always double check. Can you hand me that, Jenny? That one. In VP, I think you're supposed to wait 10 minutes.
Mm-hmm.
Okay.
So just
But she already
make
has it
sure.
changed, so.
But you can see
And yours
that
is changing
negative
pink, too.
is colorless slash
So-
pale pink. And then the
Positive.
positive is pink slash red. This one's a hard one
Number
because there's always little pink. If your result is not very clearly positive or negative either way, just make a little asterisk above it, like a star.
Okay.
And then when you come up to see what the determination is, we can always double check both ways. Okay?
Mhm.
Yeah.
Okay, I'll let them know that, because before I was told to just tell them positive. Okay. So actually, if it's questionable,
What was the last thing she said to you?
just like make sure that you
Yeah.
note it, like with a little star or
What do you mean?
something.
Like, what did
Because
she
when
just
you
tell
put
you?
it in, then it says that bacteria doesn't exist,
On that.
then you know
What was she telling you on that?
Put a little star. If it's like questionable.
Like if the color isn't like exact,
it won't hurt to know
Star
that
above it.
if it goes through the machine.
so don't put negative or.
You can, but just above it, like in this box, you're putting positive or negative, and up top, you're putting a little star. if
Just to
it's
indicate that it's
questionable.
not like
Correct.
exactly the color.
If you are doing that and there's any that are questionable, put a little
You see?
star
You heard it?
Yeah,
next
that's
to it, so that way you know if you don't get a result.
So you guys are just doing positive or negative changes?
Correct.
Yeah, and then if you're not sure on the color, put a star.
You know
above
how
it.
to read it, right? Right?
So we're just comparing this to this?
Yeah, positives
so
up
if it's
top and
positive,
negatives on
negative,
bottom.
and then let's say like this, your first one, you see how this is yellow and this is white, but let's say yours is a little more yellow than the picture, you'll just put a star on top of it, because it's a little questionable, but it is more.
Okay.
That's what she wants us to do.
Yeah, it's not.
This one is just 1%. You don't add carbs. Okay, so keep doing that. You don't forget to bring a box of these.
Okay, so we didn't have to do it? No. That would be why it's not there.
Why did we skip this already? All these
I know, I was like, okay. Let me do this. So positive or negative?
And this didn't help at all.
And then you compare. So we gotta go down the list. So see how this is like a little bit yellow and that one's yellow for mine? Then that would mean it's positive for number one. So then I would go positive like that.
So now you
That's pink and this is orange. Mine's orange. Yours is orange, so negative. Well, it looks like it wanted to change, but it didn't, so that would be my question.
The VP's kind of questionable.
So that just means it doesn't make
I'm gonna wait for VP because he said it needs like 10 minutes.
Alright, I'm gonna go back here, cause look, these all look the same to me. How's yours? Yeah, yours does have blue.
You told her, so it should be all black if it's positive. You were asking her.
I
If it's
have
just a
little black
little bit,
Yeah,
it's
it's just
negative.
negative.
Yeah, me too. Which one? This one? Yeah.
You're having a hard time with this one?
And with that, I keep asking questions and I feel ignored.
Which one's AR?
And then we're just moving forward to the next one and the next one and the next one, but I'm dead ass asking questions about the one we're fucking on, so that's just irking the shit out of me because there's more than one teacher in here.
So let's see. ARA. So that would be positive. And then Amy.
Definitely don't look
Yeah,
either
so
one
positive.
of those.
No. So looks like I got a little green.
Yeah, mine has a little green on
I'm
top.
gonna ask her about it.
Mine doesn't. I don't know.
So for the MAN,
Mm-hmm.
right, it's like a little bit, but if you look on
Yeah, for the
here, it
lines.
has green in it, but down here it's blue. So
And what's the sheet say? Because yours is like a little orange, which is a whole different thing. Blue, blue, green. I would say no change then. For you, say
it's
negative.
either negative or
Yeah, no change is negative.
that's not
Well,
even blue.
One+4.
I'm gonna ask her. See.
This one
Yeah.
would be 2.
Well,
Pick one to start with here so they can have a number.
I know SAC
Okay.
is
SAC.
yeah,
Yeah,
SAC.
that didn't
Because you
change.
have an asterisk, we'll try both.
I would
I'm gonna ask her.
lean positive, but then like put a
If you
little
guys
star next to it, like you did for the other one.
Lean positive, right? Because it's
Yeah.
definitely not blue.
It's definitely not blue.
So
So you can put like a little star. Asterisk. I can't say asterisk.
Yeah. They're both in the same number. 7, let me put this back to 1. No, this is actually provided to everyone in case you wanted to do this at home.
Mine has some
Or
blue
if you
like on
would
the
like
surface,
to leave it behind.
but the rest is
Okay.
7,
yeah. So it'll be negative.
7.
Yeah, where the black particles are.
Oh, gel is the one with the black parts, so it would be negative. VP? Mine is pink.
Yeah, mine is a little pink.
It's neither.
Maybe that one? Probably is this one. The colors are hard.
So I'm gonna do ashes. I'm gonna write pink.
Okay. So I would go with this because that other species,
I envy used it.
but let's yellow and then we'll
Mine is yellow.
your idea.
No, it's right here. It tells you if it turns super pink. Right here. It says is it colorless or pale pink. It was pale pink when she was standing here, but it just turned super pink. It was like not pink like that.
mine's very pale.
I was gonna say, it's the only one that starts with K. Oh, wait a minute, let me.
So that's
Well, there's
positive.
Klebsiella aerogen.
Pale pink is negative. Pink is positive.
So yours is positive and mine is negative.
Correct.
Mm-hmm.
VP. Oh yeah, I left that because I didn't.
Oh, yeah.
I know I skipped the ones I didn't know, so I could read this one by one.
I just know she said. I took a picture of that, by the way.
Feeling good about your sheet?
So mine's a brown. Yellowish, grayish yellow. It's a positive. Oh, grayish yellow. That's what mine is. Yes, mine's a positive.
I don't have a,
Let me see.
Mine too.
I just didn't want to put positive.
Yeah, add anything that you did get there, so.
And then mine
And what is this down
Yeah,
here?
only on this one.
Mine's
Because
yellow.
otherwise everyone has the same.
So for man, from what is yours?
It says if it's what color is yours?
Mine's yellow, so it's positive.
Yeah.
For
But
Mel,
I just noted up top because all three of these, the MAN, the INO, and the SOR, all of these should have a little bit of green in it for being positive. So it was positive, but there was no
Yours is
green.
positive.
I don't know
'cause
why
it-
I
But
it's
I
not
know,
under-
but that's why it says blue
It's
or
no longer
green,
blue.
or it says yellow or gray, so
Oh,
which
okay,
one
so
did
And
you
this
choose?
last one here is your
I put a
office
star
space
above
test.
all of them.
So man,
That number
Because it's
four.
not gray, this is green.
This one?
No, this one.
man
here.
is supposed
this
to be
one.
blue
Yeah, here.
or
For
blue
what we
green,
did
which
with
makes
the
it negative,
CSM
and if it's
plate?
just plain yellow,
Yeah,
it
for
makes
the
it
little
positive.
street thing.
So if it's
This is
negative, if
considered
it doesn't
blue.
show results.
You do negative.
Well, that's why she gave us the chart, because
Make
the
it a
picture's
positive.
not accurate, she said. She told
So-
us to look at
And then
the
this
chart.
last
So
one.
blue,
this
green.
For man is blue, it's either all blue or blue is greenish,
Yeah,
and that's
The
negative.
rest of those.
So what happens if it's all yellow?
It's positive.
Yeah,
Mine's
This one,
positive.
it has
that's what I
some
put, positive, but no green.
greenish blue.
If yours is all yellow and
Yeah, I
there's
think-
no green, then it's positive.
When you look at this thing here,
Yeah, yeah.
But
It
that's
says
why I was
that
trying to
that's
explain to you,
okay,
I
if
just
I
put that
remember
there was
correct.
no green.
because it says
No
blue
You're
or
saying
blue
it's
green.
blue.
I don't
is
even know
also
why,
positive.
because the man here, I see
So
what you're saying,
you just
but
have
the
kind of
man
a
here has
combined
green.
positive.
Okay,
Or
so
a
blue, blue,
positive
green.
result.
So like this right here where there's a
But
blue,
the
bit of blue
positive
that would
result
still be
here doesn't even
positive.
say it
Yeah.
has green, so I guess that.
Yeah,
I
because
get
that
what
just
you're saying.
could mean that your bacteria isn't
But
still
look at
going
it
or
says
attacking.
yellow for
And
positive,
then-
and
Sort of
no
split
green at
expression.
all.
For both of ours,
So I
or
guess
just
I still put positive, I just am noting
I'm gonna
that
ask
there's
her though,
no,
that's confusing, no, but I get it, but look it. It says
So
there's
I
green
would say
here, but
that's
there's
a
no
positive.
green here,
She's
so
probably
if there's
not.
green, it makes it negative, but there's only two
Because
down
if
here.
you mix yellow.
So you get what I'm saying, so I guess I'm confused when I'm telling you.
and then for the VP, ours
Now that
turned
you said
pink,
that.
Yes,
which,
very much a positive, yes.
okay.
Mhm. And
Thank
yours is
you.
positive.
Oh, I guess we're bringing all our things up, there's no time to question her, I got a question.
What's up?
Yep,
For the
and then
man,
number
it
15.
has
So
green
just
here
right.
and
Mhm.
yellow, but it says if it's yellow, it's just positive, and then it says if it's blue and then green, it's negative, but down here it shows no green, the green's up here.
I just wrote it right here. And I'm sure it goes right here, but.
So that would mean that like you have a positive for sure, looking at yours, because yours turned yellow.
No, but I like to
Or
understand the
matches.
paper, so like, which
Oh,
one would
yeah.
be incorrect, which.
So neither one is incorrect. It's just that when you have, like, say you didn't scoop up a lot of bacteria and from your little tube, that's when you get a little bit of a blue line at the top because it's just less bacteria in there.
And because it's not blue, bright blue, it automatically already is not negative.
Once you get your
Because not no
identification.
blue.
No, I know the
at all.
blue, but
You can
here
clean
it
up.
says blue and green.
This is due on
means
Thursday, plus
that
a
it's
scan
negative,
of
but
the
here there's green and here it says
API
no
20
green, it just has to be yellow, but there's green up here saying that it's positive.
Yeah.
But this
So
paper's
that's just
different.
kind of having like a weaker diffusion of it. So it has to react with all of the solution in order for it to be perfectly yellow. And sometimes that just doesn't happen. And it still shows that the bacteria has activity then.
But because majority is yellow, if it had green, it's still yellow.
Mm-hmm. Yeah, it's still mostly yellow. If it's green with a little yellow spot, definitely mostly yellow.
No, I have Eno. It says blue, blue, green, or yellow. Mine's blue, blue, green, so mine's negative.
Girl, all mine are yellow.
I don't know.
So all mine are positive.
Mine's yellow, it's positive.
It wasn't labeled anyway, so I didn't.
And SOR, if it's yellow, it's positive.
Yeah, I guess if it's a lot, yeah. Well, no, because look at
Everyone SOR?
SOR. If it's yellow, it's positive.
Okay, I got all positive.
You guys are done with your plates?
Yeah,
Let me make sure I have the pictures.
I think. I didn't really get the
He
paper,
was telling
but
me,
it is
he
what
was
it
telling me,
is.
he was like, you don't need the pictures of those because it's not necessary for it. Yeah, he's like, it's not necessary, all you need is the information. Because you're never gonna use the
Hopefully no
pictures.
one's
As long as nobody falls,
six
then
holes
you
in
fix
it.
themselves every bacteria.
Yeah.
I would try.
You look confused. You good?
Yeah, I'm just about to clean up a little bit before I bring it.
Yeah, you can throw that out if you're ready. And yeah.
And this is all we need is this.
Yeah, you can throw that out as well. If you feel good about like all your notes, if you feel like you have complete notes, then yes, go ahead. Yeah. Oh, yeah. Never, when in doubt, take a picture.
Yep. Same.
And then that's your oxidase test with the
Okay, I need a home.
which you're gonna take from your TSA plate and do that same thing.
Of which one?
Off your TSA plate.
So, yeah, your triptych soy.
No, you didn't do.
Did you guys both do it?
No, she didn't.
Oh,
You
you
did
did your
this?
oxidase test for for the, so you gotta do that too.
So get your TSA, please.
Take a picture of that.